Good. Thank you. This is Michael Miller at Johns Hopkins in the Center for Imaging Science, and I'm here with Daniel Tward and Anthony Kolosny, also the Center for Imaging Science. We're very good at being interrupted, so feel free to interrupt. We've heard this talk before, so this talk is for you, who's ever listening, and we take it as good if you have questions, and so please interrupt us. So we really are appreciative of the Exceed facility and the grant. We've been working since our history in the National Partnership of Computing Infrastructure with Supercomputing coming out of San Diego and, of course, Texas. And so this continues that work, and we're really pleased to be part of the overall NSF computing environment through this project. So we're going to be talking about our particular neuroinformatics tools in what we call MRI Cloud, and MRI Cloud is something that you can get onto. We currently have about 8,000 registered users through the original MRI Studio platform, but we're moving all the services that were in the MRI Studio executable, which was a downloadable program, still is, into this cloud environment. And so we're going to be showing you the functionality, and the back-end computing for that is almost all being hosted off of through the Exceed Computational Anatomy Gateway. So the emphasis of our talk is sort of the functionality of why people want to use services like this, and it's really oriented towards knowledge-based image analysis and this overall field we're calling neuroinformatics these days. So the general research area is brain MRI. Now, we're at a major medical school, and we're involved in lots of imaging projects, and there are lots of parts of the body that are being imaged, and we have active projects for whole body imaging and radiation treatment planning in the body with Duke, but we're only talking about the brain today. That's our principal focus here through the radiology pipeline that we have going, and it's the entire focus of MRI Cloud currently is totally focused on brain. So here are examples of brain MRIs in different modalities. They're all roughly at about 1 millimeter, half millimeter resolution. The rightmost picture, the color picture, is interesting because it's a structural image, but it's called diffusion-weighted imaging, and so it gives you not only contrast at a voxel like the first two on the left are T1 and T2, but it also gives you orientation or direction of the wiring of the brain. The bottom shows some disease associated with stroke, and MRI is used heavily there as well. So that's just to show the kind of data. So this is all at roughly the 1 millimeter scale. The current information flow in radiological diagnosis is, the image quality and consistency is maintained on the scanners in the radiology departments and programs, and we've been fortunate to be able to get them to adopt for these cloud applications and our most advanced image analysis technologies to adopt research protocols. And so currently we're able to take all of our research protocols that we have in our NIH-funded research centers, and they're being used for pediatric imaging and brain imaging in the radiology department, even though they take a little bit longer and they're not the usual protocols that have been used. They've adopted them. The data is currently put into local databases. Once the image data is put there, we really can never recall it after it's been looked at by the doctor. It's warehoused up in an off-campus site. Johns Hopkins really don't relook at the images, but of course you can do currently searches on the clinical variables that have been entered by the radiologist. And so one of the major technologies that we've introduced is the ability to search on image. So just like you can today, using face recognition with Google or Facebook, you can search on the image of a face, not just your name, but the actual image of a face, and say, oh, this must be the same person. We've essentially built technologies for taking these complicated images and making them searchable, and that's essentially the pipelines that we're going to be describing here, so that you can actually call out of the database something that looks the same. It may not have the same disease label, it may not have the same age label, it may have no label at all, but you can still find similar cases, and so that's what we're trying to augment the current radiological workflow with. To do that, we have to go beyond the unstructured image, which is just 3 million numbers, which you can't search on, or 3 million voxels, or the unstructured text fields. And so that's what we're going to be describing. I almost just hit the hang up button here on the phone. Okay, so for that, we are part of a growing discipline. We call it neuroinformatics for the brain, but image informatics at its heart is image analysis. So many of the technologies that we've built in 15, 20 years are complementary to technologies that exist, that are emerging in computer vision. Of course, biological applications and brain applications are more complex in some ways and less complex in other ways, so they're particularized. The key part of it is to solve the curse of dimensionality, just like the current focus of all machine learning today, which is to go from millions and millions of dimensions to the important discriminating dimensions that are a finite number, maybe 1,000 dimensions. So we're going to show you a 1,000-dimensional reduction of the original multimodality images, which may have started at 10 million dimensions, and we reduce to maybe 1,000 dimensions. And those 1,000 dimensions are then searchable. Of course, in computer science, this is called an index, and so it's just like indexing in speech recognition. Of course, at the heart of it is indexing of the phonetic stream into an index that you can then search and compare. And so here we're doing it in imaging. The informatics or the information becomes that index essentially.
Can you say a little bit more about for the general audience what you mean by dimensionality?
Yeah. So if you just take a 1 millimeter scan, which is they've adopted research protocols. So usually, if you go to just outlying hospitals, radiologists will look every 5 millimeters at slices that are 5 millimeters thick to sort of reduce the number of variables that they're looking at and the number of scans they have to put on the light board. And so they give up resolution, but then they can sort of grossly look at things and look at them quickly. So you might, if you were to reduce to 5 millimeters, you might only have, instead of having 200 sections across the brain, you might have 20 sections across the brain. You can put them all up on the light board. And if in plane they're 1 millimeter, then you would have maybe 200,000 total voxels would make up all the light board images. We're at 1 millimeter resolution, and they've adopted the research protocols. So for a standard T1 image, you now have 1 million to 3 million voxels of information. You'd have to put 300 sheets up on the light board, and you'd have to look every millimeter in section. So that's very high dimensional in that at every voxel location there is a number. So in a T1 image, it's the contrast under T1 density. In a DTI image, it's a vector associated with orientation, but at every voxel there's, for a vector of orientation, there would be 3 numbers. That was the red, green, blue. And so those are dimensions. So if you add all that up, you're starting with something that's on the order of 10 million numbers. And of course, radiologist isn't seeing in a 10 million dimensional space. He or she is looking in a much lower dimensional space, but it's the smart space, the right space, the discriminating space, the space that has to do with where the information is. And so discovering that and reducing those dimensions to that is, of course, the essential problem. A concrete example would be in face recognition today, even if you have a high-resolution image of, let's say, 640 by 640 or 1280 by 1280, that would be 1 million pixels. The best face recognition solutions are representing faces in a 30 to 50 dimensional space, even though it starts at 1 million dimensions. So a basis of roughly 30 to 50 dimensions, but they're very informed dimensions. They're very smart. They're carrying the important discriminating information, and so that's the idea. So the art, of course, is how to reduce dimension and how to aggregate coordinates without throwing the baby out with the bathwater. I think that's the old expression. So the curse of dimensionality is something like, you have 1 million voxels and you have n images. You might only have 100 images. The standard radiologist in their lifetime to become an expert sees about 3,000 training images. In standard training files, you might have 100 cases. So you have some finite number n of controls, and you have some finite number m. It's much less than 1 million, and you'd like to be able to extract from the 1 million voxels the information that really tells you about what is predicting a person to ultimately have a disease like Alzheimer's disease or MS or Huntington's disease or any of the diseases that MRI is used to discriminate. So how to go down in dimension from the 1 million voxels. And so we do that by essentially using the ontology or the language representing the language that radiologists use at the scale of 1 millimeter to represent the brain, and that's called an ontology. And there's about 400 structures in the brain that are seeable in MRI and people understand or track, and that's going to be the key to it. So we'd like to reduce the dimension just like when you get a blood test so that we can get a series of informative numbers. That we can't work with 3 million numbers or 1 million numbers, so we have to get a series of numbers like you have in a blood test that are essentially telling you the essential information in that scan, and then that gets put into the large searchable database along with your age, along with your clinical phenotype. Patient is reported to have sepsis and they have a high fever 3 times in the last month, that kind of information, along with our index. And the question mark represents how to go down in dimension to that information. So here's how we do it. It's not surprising. Information is stored in a hierarchy, just like in language we often associate different semantic concepts at different levels or a different hierarchy. So we essentially we reduce the dimension by representing the different brain parts at the different scales at which they're manipulated. So in one part of our representation, we might only have 11 structures representing the entire right side of the brain, which would be half of the 1 million voxels. So we would have gone down from if you look at the bottom row, we would have gone down to 21 structures representing the brain, and they would be structures like right side, left side, cerebellum, pons. Those are global names, and the representation would be associated with their contrast, so you'd have a single number for each of those substructures. If you look to the second column, we might go down to 60 structures, so we would subdivide the left and right into different lobes. You can see different colors there that represent different lobes. And in the first column, there's 254 structures, and that 254 structures is an ontology of all the cortical and subcortical structures that you learn in neuroscience that you can actually see. So that would include all the white matter tracts that you can see, all the gray matter tracts. Things that you may have heard of in the literature, associated with memory would be amygdala and hippocampus and entorhinal cortex. Associated with motor would be cerebellum and caudate and putamen. So all of those terms. So we have this ontological representation, you can think of it as a coarse parcellation, and that's what will form the index. At the finest level, there's 254 structures, so we've gone from a 1 millimeter image, which has 1.2 million voxels, down to 254 structures, but we also maintain 60 structures, we also maintain 21 and 11, and those numbers correspond to ontological terms, like if you read in a clinical record, the doctor will say the temporal lobe. So the temporal lobe is a collection of substructures. Amygdala is in temporal lobe, hippocampus is in temporal lobe, entorhinal cortex is in temporal lobe. So the doctor might say, appears that the temporal lobe is small, which would be consistent with dementia of the Alzheimer's type. So temporal lobe is an ontological term, so that's part of our coarser parcellation. But they may also say, and within the temporal lobe, we see that the hippocampus happens to be indicated because of episodic memory loss. Well, the hippocampus is also in our representation. It's at a finer scale. And so if you add up all the hierarchies, there's about 400 structures from the 254, the 60, the 21, and the 11 that are shown there. So that becomes our parcellation, and so that's how we essentially go from 1 million or 1.5 million pixels in the image, or voxels, to a much lower dimensional representation, and that essentially forms our index.
Pardon me if this is a naive question, but just wondering if it also flows the other way. That is, the ontological representation that you have, or the structures. I'm guessing some of these go back 100 years or more in medical science, and obviously predate the sophisticated tools that you're bringing to bear now. Do you see things going the other way, where you might be identifying structures that were not possible to see before using coarse methods?
Yes, and that's happening because now it's routine. We can maybe do 0.75 millimeter isotropic. It was 1 millimeter. In 17 machines, we're doing about a 0.5 millimeter isotropic. And at that resolution, for example, the hippocampus has a really nice sub-ontology or ontology that has 4 structures in it, and we know the 4 structures that you can see at the in 7T are very important, because for certain kinds of illnesses, one of the structures is implicated versus another structure. You may have heard of some of these terms like subiculum or CA or dentate gyrus. These are like an ontology that sits below hippocampus, and we expect, since we're trying to connect our atlases at the 1 millimeter scale with more refined atlases almost to the histological scale, that the ontologies will grow and will have more and more terms as resolution changes. In order to do this, we have to be able to take essentially knowledge. You could think of it as textbooks. We call them atlases, and be able to put them into the coordinate systems and apply them to the individuals. And so we have to do both knowledge creation, which is generate atlases that have these ontologies in them that are age-matched and disease-matched, because that's very important. And we have to then be able to apply them. Each person is different. Everybody's brain is a different shape and size and locally varies. So we have to be able to apply that knowledge. And then once we can do that, we can then have an individual index for each person's brain and look for what's common. Just like that for speech recognizers, they had to solve the problem that some people talk fast, some people talk slow. Even you yourself, if you've had some caffeine, you'll tend to speak more quickly, and then as you get more tired, you might slow down. So there's variation in all of these kinds of human signals, and we have to deal with that. So the basic overall plan, what we call MRICloud.org, is you have this high throughput imaging centers that are generating MR images. You have a service. You send the image to our MRI Cloud. As part of the service, there's a knowledge database of annotated brains of similar age and disease. Those are applied to understand the particular individual, and then an index is created. Here it's shown as a stock sticker chart, which every row is a different imaging modality, and each entry in a row is one of those 400 structures. And if you have available statistics, then you can take a modality and a structure, and you can ask, how does this structure in that modality compare to a big database? And for the parts of the index that are a large deviation, either small or large or a very different contrast, you can annotate it, and here we show in different rows, bright red, which means 3 standard deviations, and less red might mean 2 standard deviations, and you now essentially have a way of starting to score automatically and build indexes that have statistics built into them, and you can now load this into your database and then come back to the database and query it. And so that's the idea of the cloud. And so now I'm going to show you a little bit more about how this works. So how do we do the data reduction? I've told you about the ontology of 400 structures. So first we need knowledge about structure definitions to teach computers how to reduce subject-specific information. So we look at the same brain under all these different ontologies, and then we look at many brains, and we are currently annotating the ontology shown on the right. It's a hierarchical ontology, so if you were to go to MRI Cloud and click on any of those black or red squares, like if you were to click on, let me put my glasses on here. If you were to click on frontal lobe, you would see frontal lobe breaks into SFG left, SFG right, PFC, prefrontal cortex left, prefrontal cortex right, polar, and various substructures. So the whole thing is hierarchical, and we're generating, to give you a perspective, we're online now with somewhere between 10 and 20 atlases, but we're generating over the coming year about 270 atlases that will be matched across age from 1 year to 80 years and will be oriented towards the diseases that we're studying at Johns Hopkins. So that's the content that we're creating. The next thing we need to do is then apply the content in those atlases. Those are like the training records. Every radiologist, when they get trained in a particular area of the brain, they have training data that are particular clinical cases that have been annotated that they learn from. And we're creating these atlases for the machine to have. So the next thing we need to do is to apply the knowledge that's stored in those atlases to individual MR images that come in from the cloud user. And so that's the central computation that the Computational Anatomy Gateway is providing. And a term for this is multi-atlas segmentation, essentially, or multi-atlas parcellation. And this is exactly like parsing in speech recognizers, only here you're parsing into this ontology images rather than word streams. And basically, we're coming online with, as I said, 200 atlases. Currently, we make available around 10 to 20 atlases. A person sticks an image in and a 3D volume, and that's shown in the center. And then each atlas is applied to that image in the center. Each atlas is used to interpret the image in the center, and then at every voxel location, so at the finest resolution, the voxels is about 1 million voxels, so at every voxel location, a decision is made as to the ontological label, and of course one has to understand what the voxels around you are doing, so that's all part of the algorithm. And the complexity of that is maybe I should have had the number here to give you a sense, but let's say on a 4-node, on one of your 4 nodes, I don't know, 4-node is 4-node a basic unit on on an exceed cluster, or like on our cluster here, like a basic unit?
Well, usually each node has 24 cores.
Okay, good.
So, yeah.
Okay, so let's say on a 1 node. That's great. Just to give you perspective, we're not, we have exact timings in all of our ports and everything, and we're going to be talking about the GPUs at the end here. So 1 node has 24 cores. It might be, if we had 1 atlas, that the computation would take 1 minute on that order. So it's still not really real time, but the whole service is asynchronous, and the user gets back that the job is done, and then they, and it's available to them on the client side. For 15 atlases, it might scale linearly because of the way we've implemented it, and so if we have more than one node available, we might stay close to the 1 minute. On the average, I think our throughput for 20 atlases is about 5 to 10 minutes, 15 minutes on the Exceed system at Gordon, including data transfer, getting it back, and that kind of thing. Yeah, I think there was a question. Okay, I'll continue. That's roughly right, including data transfer. So like 15 to 20 minutes per brain, right? And some of that is the queue and all the upload times and all those things. So once we do this, we get out this, this Wall Street ticker tape here on the right. You put your brain in and you get out, in yellow is every region. There's 400 of these regions, and you get out the volume and other statistics associated with different modalities. So T1 may give you volume, but DTI gives you 3 orientations, and T2 might give you a contrast associated with stroke and various things like that. So each column is a different modality, and clearly we haven't yet opened the service for any multimodality submissions, but we're about to come online with the service for DTI, which DTI is like 3 to 6 modalities at once. So the computation time is likely to scale to get a little longer for DTI. We currently, in the executable program, MRI Studio, many of our users do DTI. Almost all of them do DTI. So we think the DTI Studio will increase our traffic quite a bit. So we now have this vector of numbers on the order of 1,000 numbers for, let's say, 1 or 2 or 3 modalities, and that's basically our index. So for every brain now, we have this, and so here there are 4 rows, so that corresponds to different numbers. T2, fractional anisotropy, MD, volume, these are different features. And the colors correspond to large deviations away from normal. We're collecting normative databases as well as disease databases. And so each one of those 3 rows is a different kind of grouping. So we have focal dementia, we have diffuse dementia, we have normal anatomy in our geriatric group for all of our large studies that we have data for. So now it's, I guess, pretty clear that we've essentially now reduced these images to an index that we can do all of the classic machine learning that's being done and developed in the world, and we're doing that, and you can think of that as knowledge mining. We have a large team of radiologists here working on simply annotating the clinical record. We have collected now about 7,000 or 8,000 pediatric cases through our radiological workflow. And so they're being annotated for about there are about 100 diseases that kids have that bring them in through the pediatric MR neuro clinic. And then we also have a huge geriatric, we're a major site for study of various dementing illnesses. So we're annotating those with the clinical record, and then that forms training data that we can then do machine learning on. So once we have a brain and we've structurized it into quantitative matrices, then we can search against past similar cases. So we're building a tool that we're putting as a client down in the radiological workflow area that allows radiologists. We currently have 4 radiologists that are trying to use this now. They can take the image on the left, they can structurize it, and then they can go into the database of log patients and say, independent of the clinical record, I know this is a woman, I know it's a 44-year-old woman, and I know she's had temperature 3 times in the last 2 weeks. I'd like to know what other cases in the database, in the training database that's been looked at are similar, because I'd like to see what previous data is saying from other doctors on the floor. So we're using this for training, and we're trying to see if this is a value proposition. And so what that then does is bring up other similar cases, and then you have, since they were training data, you have available all of the clinical record from very highly trained, more senior radiologists that have worked over the last 5 years in the clinic and seen rare cases, so you'll see things that are similar that are rare and that sort of thing. Also, we're investigating whether this is a value proposition or not to assist in terms of diagnosing. So many illnesses are diffuse. You know, we think that one of the reasons why schizophrenia is so difficult as a developmental disease is it's likely many things, and in the future we may understand what those many things are. You know, when people often use the term that we use for light, broad spectrum for that kind of thing. So what we're seeing that, of course, with dementia as well. We know there are maybe 12 to 15 different variants of dementia here at Hopkins that people are studying. And so here's an example where we took 100 cases, we put them into a dimensional space of about 100 dimensions. So that meant we took a 1 million voxel image. We first turned it into our 400 long feature vector. This is just T1 images, so it's a single 400 long feature vector. We then do machine learning on it. We go down to about 100 dimensions. And then in those 100 dimensions, we show all these cases in the first 3 coordinates. And you can see that we're able to cluster, we're able to maybe get sensitivity and specificity down to about 80% or 82% on a data set like this. But there's definitely overlap. So if red is among greens, then what that means is that the semantic variant looks like the controls, because the controls are green, and we put them in the lower left to show outliers. And if you look at these brains, you'll see in what sense they're similar and in what sense they're different. If you look at the semantic variant on the right, you see the semantic is associated with temporal lobe, and the temporal lobe dementia there is circled in yellow. And so those are all common, and they're showing up in the right place in the clustering algorithm, because in one of the dimensions, that's signaling what's happening in the temporal lobe. So that's the idea. I think you get a good idea of the way that we're trying to use this kind of thing. We wanted to have a couple snapshots to MRI Cloud, and so, Anthony, I think I'll just stay on this and just present this. Is that okay? Anthony put this together to give you a sense of what users get to see. So they log in. We currently have it so that you can be an MRI Studio enrolled person, or you can come in through other kinds of credentials. But almost all of our users are coming from the fact that we already have an executable program that users are hitting and have been hitting for years now at a rate of about 10,000 brains a year, roughly about 1,000 brains a month. And so we're making our first market to try to get as many services as we can in the cloud environment for those users, to try to get them to be using the cloud environment. And we're starting to include in our MRI Studio tutorials, which we do at all the major medical imaging conferences, we're now starting to include MRI Cloud since in January we launched, and it's now fairly stable. So shown on the left is one of the input screens. Shown on the right is an output screen of when the job is ready, it's been run through, and it's available for the client to visualize results. The screen says that in the visualization client, which is a very thin client, we have 2 kinds of visualization. We've been working for a long time with ParaView, and we have a very advanced, heavy-duty, non-client-oriented program called CAWorks, which uses the ParaView platform, and we've been developing that with Kitware for years. It was part of our work with the Burn, which of course involves San Diego and supercomputing, and you guys for a long time on that. But we also have a very thin client that we give users, allow them, and that's what this screen is showing, associated with all of the structures, segmented structures. We also have triangulated meshes. A lot of our shape analysis, and you'll be hearing from Daniel about that in a few minutes, uses triangulated meshes. So we do a lot of meshing. And so in the upper left window, that's a 3D object, and we've essentially written a very thin client that runs totally in software so that it can run everywhere for 3D sort of graphics. That's a manipulatable object in 3D on the left, and the other two are sections, and it shows the results of the automated ontology injected into the three views. And then to give you a sense of how we're using data, the rightmost picture showing little green dots is all the cases that have already been uploaded, analyzed. In this case, the user is looking at peripheral frontal white matter, and then what's shown is the particular brain that the user is looking at as a red X, and you can see that the age is about 63, and the volume is relatively large relative to the rest of the data in the database. The red X is very high for that particular feature. So this is the kind of functionality that we're offering. Anthony made a nice chart that shows, where we've been since July 15th, 2009, and I guess I should look at the right as 2014. So we're currently, but that goes, that chart only goes to January 1. It doesn't show, right?
Yeah.
Okay. So that's the kind of traffic that we've seen historically under, before we connected to the Exceed and opened the gateway, we have our own supercomputer here, and MRI Studio was hanging off of it, and that's sort of the number of brains that we've processed historically, showing you the sort of, for that number of users using MRI Studio, that's sort of the number of brains traffic. I guess we're doing now through MRI Cloud, when I look at the current list every day, like 20 or 25 or 30 a day. It's roughly this 1,000 a month thing now. We could give them, actually, you need a password for them to see that local list. The list that I always look at?
That's our internal log.
Okay, that's our internal log.
Okay,
so we have an internal log that allows us to see the traffic to MRI Cloud, and I guess this chart doesn't show that, but I would say that we're not yet in steady state in terms of use, although we're doing like 25 to 30 jobs a day in the MRI Cloud. So this is a chart that really shows the service, but when it was being hosted solely on our own computer, and it was a much simpler service. One of the things that we've been able to do because of the Exceed platform is historically we only offered one atlas, so we were only offering it for sort of control, middle age. Now that we have 15 atlases, the complexity is about 10 to 15 times higher, and so all the jobs that we're running now are that much bigger, and so they're all being done in the Computational Anatomy Gateway. Okay, so I was going to turn it over to Daniel, who is now working on many of these algorithms, but trying to integrate them to GPUs because the dimension is getting bigger. We're seeing that we want to be able to do more things. So if that's okay, he'll just continue the webinar. Is that okay if we just continue?
Will you take a bit of question before you switch over?
Please. Yeah, we want you to talk to us.
Okay. So it sounds like to me you guys are dealing with personally identifiable information. Can you talk a little bit about how you handle the HIPAA compliancy?
Right. Yeah. So Susumu's not here, and so he's the professional radiologist who's in charge of all this. But Anthony will tell me if what I'm saying is wrong, I'm sure. So basically, we're forcing all the users to, we're not, for outside of Johns Hopkins, we're not accepting anything directly out of the PACS. So all of our users, we basically have a nice little simple program that's very thin that the user can download if they don't currently have the data that comes out of the PACS already made into analyze format or an equivalent to analyze format. And in these analyze and equivalent formats, there's no information at all that that allows these submitted images to become then personally identifiable. And so for everybody outside of Hopkins, outside of our sort of security setting here, that are using the MRI Cloud, we basically force them to keep all of that on their side, and we don't have any of that on our side for any of the data. However, for all of, one of the other things that's going on, which is all of our research projects in which we're all HIPAA compliant and trained, and CIS is integral to the, we have several program projects here with the scanner. The scanners are all at the medical school, but we do all the image analysis on the Homewood campus, and so that's all very heavily firewall protected, and for that it comes directly out of the PACS, and it gets, of course, archived. We don't intercept the radiological workflow, so the radiological workflow continues. It all gets sent up to Mount Washington, that all exists. But for our project that we're doing with radiology or any of our funded NIH projects, it goes right out of the PACS, it comes to us. We handle all the de-identification, and then we're basically storing all the data in XNAT. We're using XNAT as a databasing utility here for that data. So we're trying to avoid HIPAA issues except on all of our funded research projects. So then, for our research projects, we, of course, have our committees here at Hopkins that enable us to work with the data and follow the usual rules of compliance.
Okay, and so I'm wondering if the XC resources you're using are dealing with any of the HIPAA data, or is it only dealing with the anonymized data?
Right, only the anonymized data. That's right.
Okay. And can you talk a little bit about how the MRI Cloud handles written or annotated diagnostic information, or does it handle that?
Yeah. So we were just at BISD presenting a webinar on this. It's interesting. We should get people on the call that are interested. We're funded under some of the BD2K stuff, and there are a lot of people interested in this whole informatics thing now, as you imagine, as you are. And we were just at the NIH talking about this at the National Library of Medicine. Maybe we could get the names of some of the people on the call that are interested, because then when we have another one of these at NLM, some of these things are coming up. So,
yeah, just on that note, I'll send out a list of all the people who called in today.
Yeah, and so as we said at the National Library of Medicine, we've really put a huge effort not into EPIC, but into MRI image analysis and this whole data indexing and atlasing thing. And we're working with other people. So at our place, the whole hospital is going to EPIC and using EPIC. And so we've really stayed close to our knitting. we literally have 4 radiologists who are, even though we have the entire EPIC record, it's all available, who are part of this project, who are literally recreating the clinical diagnosis again in a NoSQL modern working database that will allow us to search on the clinical record at the same time as we integrate our MRI index to the NoSQL database, which is where the MRI index goes, so that we can do querying. While in parallel, the whole effort at Hopkins proceeds on the EPIC front. And so that means that our 4 radiologists are reading the unstructured record again. We haven't invested into any linguistic recognition tools. We haven't gone in that direction yet because we've wanted to use we got one of the first versions of the BD2K to do this project, and it was relatively small, and so we have not put resources yet into more automated tools for the clinical side, and we're literally using our clinical manpower. We've created a lot of forms and utilities that allow them to annotate and put information in. We've defined about 100 to 200 fields that we think are important in this setting, and they are now searchable, and we're going case by case in doing that. We believe that soon we will be working harder on the unstructured language part so that we can actually integrate more carefully and not have to have, sort of recreate the wheel, reread everything. Although even with that, you still have to go back and quality control everything if you use an automated tool. And we're going to be integrating with the resources at Hopkins that are on the EPIC side. However, on the EPIC side, Hopkins is just bringing this massive system up, and it's very complex right now, and so groups are trying to figure out when research efforts should be putting resources into the integration with EPIC. So that's a complicated response, but we're trying to work it to be maximally efficient on our side and not spend lots of cycles in places that we can't, really influence. Should we continue? Is that okay? Are there more questions?
Sure, go ahead. Thank you.
You're welcome. Thank you for asking.
All right, I just wanted to spend another few minutes talking about another service. Can everyone hear me all right?
Yes.
Another service we're offering through the Computational Anatomy Gateway, where instead of working with individual MRI images, we try to understand anatomical changes in parts of the brain on a population level. And I'm going to mention a little bit how using Exceed resources has enabled us to build up big populations, which is helping us improve technology, and the kind of computations we're doing on the cluster that we're trying to accelerate with GPU and MPI and multi-core work. So the typical usage through this service is that a user will start with a population of anatomical definitions that have been defined from medical images. These could come from the type of parsing that Dr. Miller was talking about, or it could come from manual specification of structures. In order to analyze these structures, we start with a given template. So you can see on the bottom left of the figure there's a cyan-colored hippocampus. Dr. Miller mentioned that the hippocampus is a medial temporal lobe structure, which is important in Alzheimer's and various dementing diseases. So the tools we use will construct a transformation which links a template piece of anatomy to an individual's, which we call target, an individual specification of their anatomy. And these deformations are created in such a way that we can define distances between anatomy rigorously. So the way that I'm describing here, on the top you can see there's a flow of time. On the second row you see at each time we construct a velocity field in analogy with fluid dynamics. In the third row, you can see we're integrating the velocity field to calculate deformations at every point in space, and we can use those deformations to make quantitative statements about the anatomy that we're studying. One of the most important parts of this analysis is deciding what we're going to use as a template, the cyan hippocampus structure. And we really want our template to be specific to the population we're studying. Earlier we mentioned using 15 or even more templates such that the individual ones can be more specific for individual cases and increase the power with which we can interpret things. So the basic idea is once we define distance rigorously on the space of human anatomy, we can estimate average of populations by simply minimizing a square of some distance. And these calculations are done now on the Gordon cluster, I believe, where mappings between a template and each individual in a population are spread out onto different nodes and different cores within a node. And that information is combined back to adjust our estimate of what the templating is using a simple map-reduce operation through MPI. So this allows us to scale up to large populations without wasting a lot of time waiting to analyze one member after another. After we do this type of analysis where we compute a mapping from a template to each target, we compute measures of local atrophy and growth at each position within the template anatomy. And this data is output for users to use to do statistical modeling and hypothesis testing on their population. So what I'm showing at the left-hand side is an example of temporal lobe anatomy. The entorhinal cortex, if you can see my mouse, is the medial structure here. The hippocampus is a larger structure here. The amygdala is the most anterior structure over here. And we're looking at local change in volume or surface area over time between the bottom figure and the top figure is increasing in time, and we can analyze differences between different disease populations, and we've been able to see how the disease progresses in Alzheimer's through the entorhinal cortex. Now, as the population size has been increasing with what we're studying, we're able to learn a lot of information from these populations. So one thing we learn is empirical models of biological variability. We can understand what types of anatomical shapes are more likely versus less likely. We can understand the variance in MRI images, where images are likely to be noisy and where they're likely to be more trustworthy. And we're able to understand uncertainty in anatomical definitions. So once we start getting down to very small scales, the way structures are defined start to become ambiguous. Different groups will have different definitions. So what I'm showing on the left here, if you look at the red bumpy isosurface, this is an example of some segmentations that are created of the hippocampus by a tool called FreeSurfer. And the green surfaces that contour these but are able to average through the noise is an example of how we've been able to use these models of variability in biological shape and imaging system noise to improve the reliability of the way we analyze this data. So on the bottom, I'm showing two plots. On these plots, I'm showing the distance between anatomical definitions from one scan to the next. And on the left plot, we're just looking at scans within a few minutes of one another within the same visit. And on the right plot, we're looking at the difference between scans that come a few weeks away. So using these tools, you can see on the left side, we reduce the variability from red to green to about the level of 1 voxel within a single visit and less than 2 voxels between visits. So we're able to increase our accuracy a lot this way. These types of algorithms we're using are often iterative. So typically, given a population, we can estimate uncertainty or estimate something that depends on all the data in our population. We do a computation, and then based on what we've learned now, we repeat the previous. These expectation maximization algorithms, they're called. We're working now with multiple images together, as well as triangulated surfaces when we're analyzing anatomy. So a lot of work has gone into speeding up computations. And the work we've done has involved multi-core and GPU computing with OpenCL. We're using the Tesla cards on the Stampede cluster, and we're hoping to get into multi-core computing and accelerators with OpenCL on Gordon. And as well, we're using CUDA on the NVIDIA cards to try to speed things up. So
I'm curious, pardon my interruption, I'm curious, are you working with anybody on Exceed for help with that optimization?
No, we're not, to tell you the truth. Just trying to get our programs and libraries hooked up, but we haven't linked up with an expert.
Yeah, I don't want to interrupt all your fun doing that, but just so you know, there is whenever you're ready, or if it's useful, you can have extended collaborative support to support those kinds of code optimizations and problems like that.
Yeah, that sounds great. That's definitely something we should look into.
This is Miller again. I have a question. It's going to be for Daniel, but you're causing me to ask the question. So I thought we couldn't run our GPU codes on Gordon. We weren't able to. That we had that small allocation still that we were using in Texas Stampede. Am I wrong about that?
You're correct.
Okay.
But because OpenCL is intended to work on different architectures, the same code. So I've been working with someone in Gordon who says that we will soon be able to run it on the CPU in the Intel environment.
Oh.
So that's something that we hope to be able to do. And then the parallelization can take place. The parallelization can take place very efficiently on a single CPU or multiple cores in that context.
Also, this is Mark Miller. The Comet machine that we just put up has very fast GPUs also, so that might be something to keep in mind for your next allocation.
On which cluster? Can you say again?
It's Comet.
Comet.
Mm-hmm.
We'll consider it.
Thank you.
Yeah.
Yeah, I'll add. Comet and maybe Wrangler, a new machine at TAC, might be interesting for you going forward. And then also in the near future, two other machines, probably of those bridges will be the most relevant. There's also another machine called Jetstream. They'll be here at IU that will be available beginning of early next year.
That sounds great.
I think you're at the end of your talk. Right?
Yeah, I think we're at the end here. I just wanted to mention that depending on the type of data we're working with, we tend to be able to see orders of magnitude improvement. So working with GPUs is going to be really important for us.
Well, that's what your chart shows, right? The green is GPU, right? Okay, so I guess I just hurried Daniel along because I knew that I had something at 1 on Eastern time, but I wanted to leave enough time. I don't want to leave. I'm hoping, is there more that we should talk about? And we really appreciate your listening to us. It's really, a pleasure for us.
Yeah, our pleasure too. It was a very interesting talk. I know you have to go. I guess I had some questions scribbled down. I'd like to see if anybody else has something first, though. I don't want to keep everybody else too long either, but one question I had ultimately was, wearing my Exceed hat. What are there problems or other things that you have that you wish other people would solve for you from Exceed? Can we? So there's a whole group of people, some of whom are on the call, that provide extended collaborative support, and there are resources available to you to help with things like moving your codes to different machines and those sorts of things. Would any of that be useful for you?
Well, we're going to be writing our new grant soon for the following year. So I don't know if there are certain people that would, be good to contact as we're writing it up so that they could provide the suggestions.
Yeah, definitely. Probably not anyone specifically on the call right now, but I think we can connect you with the right groups.
Okay, so as we write up the proposal, then we could send it to you or someone else, and then you could probably know who the people would be that could work with us in order to get the machines that would best fit our needs.
Yeah, absolutely.
Okay, good.
I think it would be great if we could work with people at Exceed, because like Nicholas also would love that.
Yeah.
You know, we just hired a new faculty member from École Normale in Paris in applied math, and he's very strong in math and these algorithms, but he could also benefit, besides Daniel, from having somebody that, you know, optimizes his codes, because he's really trying to publish, you know, a lot. So this is a great connection, and historically, as Anthony can tell you, our original mapping codes that this is based on was all developed with San Diego supercomputing people originally. That's who Faisal worked with, right?
Yeah.
And then we worked with Intel.
I mean, you know, we've had great history with everyone we've worked with at the Supercomputer Center. So a little bit of the evolution is that we then worked with Intel to optimize our code. So we used the MKL and various things there. And then when we were porting it to Texas at Stampede, we had an engineer work with us for 6 months there, and he helped us with the compiling so that we were able to utilize the mic processors in the hybrid mode. So we got like a 30% improvement there. And then, you know, then we moved to Gordon, so we recompiled again. But with each generation of processors, we've been in pretty good luck, because, you know, we've been able to align with the Intel optimization through these generations. And, you know, now with Daniel doing some, you know, GPU acceleration, we're on this process again. So, you know, we're definitely
really appreciate it.
Yeah, we definitely appreciate a couple engineers to help us along.
Yeah, well, you've already gotten it, and yeah, these are exactly the types of problems that Exceed is there to help with.
I do have one quick question. So it looks like the MRI Cloud can handle 2D images, and you mentioned the 3D volumes in your second half of the talk. Does the MRI Cloud actually handle the 3D volumes, or is that a separate application?
Right. No, actually, I may have confused us with the 2D. I think we showed some 2D images, but everything is 3D. Everything is roughly 1 millimeter isotropic, or we convert it to 1 millimeter isotropic, all 3D. Everything is 3D, so all the times I was talking about, and all the atlases are 3D. When our DTI processing comes online, that's more 2 millimeter isotropic if you look at the modern sort of DTI protocols. And then the fMRI, once we get that integrated, that will be more 2 to 3 millimeter isotropic. But it's all 3D, and we do it all in register with the atlases, which are all 3D at 1 millimeter isotropic.
Okay, thank you.
You're welcome. So I just want to thank you again. This is very helpful for us. We appreciate this, and please ask us, you know, if anything comes to mind, other conferences or any venues where we can help, or chime in or write a letter or anything. Just ask us. That's, you know, we're good at that, and we really appreciate the support.
Great. Thank you very much.
Yep.
Thank you very much.
Okay.